Okay. Hello. So Tanner isn't here this weekend, so it's just me making this recording. He's out of town doing something. So we didn't have the chance to go over group problems together. So I'm just going to talk about them, any issues that I had. And then I didn't do number 5 for some reason again. And I'm thinking it's because I saw somewhere that I wasn't supposed to do number 5. Either way, I did it on my whiteboard. I drew out the DNA replication process. And so I will show that I did that too. It might not be 30 minutes long just because I don't have anyone else to talk to. So sorry. Okay. So group problems. The sequence of complementary DNA strand. That wasn't hard at all, especially since it's just complementary DNA and it's not RNA. If it had been RNA, then I would have changed my T's to U's. Indicate strand polarity, and so that one would be flipped from what they had originally. Let's see. Knowing the structure of DNA help you hypothesize a model for DNA replication. Let's see what I wrote. I wrote that helping to understand the structure of DNA helps me to understand why it twists. Yeah, I'm pretty sure I got that right, so I'm not super worried. Let me. Oh yeah, the 3 possible mechanisms for DNA replication: the conservative model, the semi-conservative model, and the dispersive model. I hadn't actually learned about this before, so this was interesting to study about. That wasn't too bad. Number 4, where it says, what would be the result of a single generation if DNA replication uses the conservative, semi-conservative, and dispersive models? I think I got this one wrong. I'm not entirely sure. I can't, to be honest, I can't remember. I said that with the conservative model, you would have equal amounts of nitrogen 15 and nitrogen 14. Just because the entire copy of the DNA double helix is being made. In the semi-conservative model, I had one strand being nitrogen 15 and one being nitrogen 14, and then in the dispersive model, I had a mixture of N15 and N14. And then in the conservative model for the second generation, well, for 2 generations, I had the DNA would be incorporated with N15. The semi-conservative model, there would be N14 in the 2 daughter strands, and then in the dispersive model I had, there would be both N14 and N15 in the parental DNA molecule. If I remember correctly, though, it was worded a lot differently than how I worded it on the group problems here, and so I was also trying to figure out if I had it correct or not, so I'm actually going to go and recheck that really quick. Let's see. Let's see, okay. For the first generation, it says 2 bands of equal intensity would be observed in the test tube. One band would be at the heavy position and the other band would be located at the light position. So I think it was talking about the mass of nitrogen-14 versus nitrogen-15. It was either the mass or it's not the density. But I would, yeah. So I wasn't thinking about like the bands on the DNA in test tubes specifically. I was thinking just about the different strands, like um, let me look in the notes really quick. Yeah, here in the notes it talks about bands being formed. Let's see. I think when I was doing this, I was looking at the experimental steps instead of the evidence for the semi-conservative replication. And the different models that it talked about. I'm not sure why, because I remember reading this, but I don't know why specifically. And so I talked about like the strands incorporated in which of the strands, but this talks about density. So I sort of got that one wrong, I think. Number 5. Well, actually, I'll skip to number 6 really quick. Number 6 and number 7. These ones were fine. I think I just missed, oh wait, naming the functions. Oh wait, no, that was fine. Yeah. Yep, we're good. Okay. So, number 5. It said to draw a linear double-stranded DNA molecule, indicate on your drawing the polarity of the parental strands in an origin of replication. All right, so hopefully you can see this. I have a really big whiteboard for studying purposes. It's kind of hard to see. Let me see if I can. Something else. Oh, here we go. That works. You still can't see it very well. So on this question, when it said double-stranded DNA, I don't know why, but this like kind of confused me. For whatever stupid reason. So 2 strands of DNA split apart, and I could not remember how they split apart, and then they replicate themselves. And so we have 2 leading strands and 2 lagging strands. We have origins of replications here and here. On the bottom, I labeled things better than I labeled them on the top. But I also have the polarity labeled with going 3 to 5 on this top strand, to 3 on this bottom strand, and then it being replicated and being and creating the complementary DNA for both of them, as well as on the top. We have helicase here separating the 2 sides of DNA. We have RNA primase, RNA primers, these Okazaki fragments on the lagging strand. And then we have our leading strand going down here with RNA polymerase creating it, as well as single-stranded binding proteins. I think that's what they're called. Also DNA ligase, connecting everything and finishing that up. And so all of this is demonstrated down here as well as up here. I didn't have it like connecting back together just because my whiteboard isn't big. But yeah, let me see it. Let me make sure I did everything I was supposed to do. Indicate the replication forks, 2 leading strands of Kazaki fragments, and products of replication indicate polarity. So I indicated polarity. I didn't technically draw the end products of replication. I mean, what are the end products? The end product being a complementary strand being created, because I did that. What I didn't do is I didn't connect the strands back together. So yeah, hopefully that was good enough. If you want to take another look really quick, this is what I drew. Hopefully that was right and I did it correctly. If not, I'll work on it some more. But so that was this week. Going well. I've talked to Tanner. I just we just weren't able to meet to for this video. So sorry that this isn't over 30 minutes. Hopefully that's okay, just because I don't have anyone to like bounce ideas off of. So thank you though. I thought I'd make this video anyways. All right. Bye.